Okay, so this brings us to the second lecture on cell signaling, and I'd like to start here by discussing, just very briefly going over the concept that we talked about. There's extracellular signals. The extracellular signals, it can be gases, they can be hormones, they can be ligands. Ligands are, a ligand is anything that binds to a receptor. That can be polysaccharide, a protein, a glycoprotein, right? So any of those extracellular signals can result in intracellular signals that lead to the activation of targets. And the targets can be things such as turning on gene transcription, changing the cytoskeletal elements, changing the metabolism of the cell, virtually anything you can think of. All right, and we already talked about different extracellular signals. We talked about gases, right? We talked about nitric oxide and smooth muscle relaxation. We talked about hormones being able to cross membranes and bind to intracellular receptors such as the androgen receptor. And now I'd like to just briefly talk to you about the hormone insulin and how it works. This figure really does show you that the activation of the insulin receptor by binding insulin causes a multitude of activities in the cell. If you look on the right-hand side, you see the insulin receptor with a ligand bound. That ligand binding leads to the activation of a number of different molecules. Right off the bat, you see P85 and P110 just below the insulin receptor. The P85 and the P110 together make a particular kind of phosphatidylinositol 3 kinase, a PI3 kinase. That PI3 kinase is going to lead to the activation of additional molecules, and all of this is leading to a downstream function. So what have we talked about in the past? What happens when the hormone, why is insulin produced in the first place, right? Insulin is produced in an effort to reduce the level of glucose in the body, in the bloodstream. Glucose is produced by beta cells of the pancreas when it recognizes that your blood glucose has gone up. You eat a Snickers bar in the afternoon, all of a sudden you have a lot of sugar in your bloodstream. The pancreas starts pumping out insulin. Insulin goes to your fat and muscle cells and it says, hey, we need to reduce the level of sugar in the blood. It's not healthy for us. And it upregulates GLUT4, which is the insulin-specific glucose transporter on fat and muscle cells. So when you look at this fluorescence micrograph of a cell that has GLUT4 in it, so this is either a fat or muscle cell. I perceive this as a muscle cell because I think it has multiple nuclei. You can see very clearly that the green fluorescence is in a lot of places, okay? You can see somewhat of the edge of the cell, but you can also very clearly see that there's a lot of it intracellularly. Let me see if this will play. This won't play still. But when insulin binds to the cell, all of a sudden, all of that, the green fluorescence will reach to the outside of the cell. So, and that is the movement and the fusion of GLUT4 containing vesicles with the plasma membrane, and now GLUT4 is on that plasma membrane and allows for the uptake of glucose from the bloodstream. So that process is really important, right? So if you don't do this correctly, you have diabetes. Diabetes can kill you. It could cause blindness, can cause you to end up with amputations because of reduced blood flow. Glucose, too much glucose in your blood, bad for you. Okay, and this is just showing you the actual trafficking that occurs. So the insulin binding to the insulin receptor, shown on the bottom left of this picture, causes the activation of an insulin responsive protein that activates the P85, P110, which is a phosphatidylinositol 3 kinase. That activates another molecule called PDK. It's another kinase, which activates protein kinase B. All of this is a kinase activation. So you have something is happening to cause the upregulation of protein kinase B. Protein kinase B goes and activates another molecule called AS160. That activates and allows for the transport of the GLUT4 containing vesicles from the GLUT4 reservoir to the plasma membrane. You can see that there's GLUT4 is actually, I don't know if it's a 10-pass or a 12-pass transmembrane protein, but what it does is when it reaches the plasma membrane, right on the far right of this, it allows for the extracellular uptake of glucose into cells, and that's going to lead to either the production of more fat, or it's going to be used in muscle cells to cause the production of energy. So, this is a very complicated process, and most of it is regulated by kinases that phosphorylate the next molecule down. There was a phosphatidylinositol 3 kinase that phosphorylates a phospholipid. That activates protein phosphoinositide dependent kinase, which that just means there's a kinase that's activated when you have PI3P, so the phosphate on the phosphatidylinositol at the 3 position. That phosphorylates protein kinase B. Protein kinase B is now active and is going to phosphorylate AS160, and AS160 interacts with Rab4 on the GLUT4 reservoir to produce a vesicle that will then be transported to the plasma membrane and fused with the plasma membrane. And that's regulated by Rab4 and snares at the plasma membrane. Okay, so that gives us an idea of different kinds of extracellular signaling molecules and the activities that they could regulate. Okay, there's another whole class, right, of molecules that just bind to receptors, and that binding, right, is bound to cause a conformational change, and the conformational change then leads to some sort of signaling cascade. So the receptors themselves can have enzymatic activity, or the receptors will then cause the recruitment of molecules that will somehow cause an enzymatic activity to occur. Most of the time, this is regulated by some sort of phosphorylation event or activation of a GTP ACE, so in the case of G protein linked receptors, or it even can activate ion channels in some cases. I didn't talk much about ion channels. I don't expect you to know much about ion channels, but ion channels sit on the membrane, right, and they bind to a ligand. The ligand causes the opening of the channel, and then ions can go through. And right, so what we're really talking about is a lot of times these ion channels allow for something to go through, a calcium ion to go through. That calcium ion, once it's intracellular, can then cause all sorts of signaling events. All right, so you can regulate other channels by the influx of calcium. We can upregulate second messengers such as cyclic AMP production, cyclic GMP production, the phosphorylation of phosphoinositides. So all of these things are going to then, right, do the domino effect of activating the next molecule in the line. Some of those are very freely soluble and they can diffuse throughout the cell. Others are actually bound to the membrane and can only activate molecules that are recruited to them, usually through one of the binding domains that we talked about, either SH2 domains, SH3 domains, or pH domains. Okay, so it just depends on what receptor, what molecules get activated downstream from that. All right, I'm going to skip that slide because that's just a dumb slide, and let's talk about G protein linked receptors. Okay, G protein linked receptors are receptors that have the ability to bind to a ligand, but then there's a G protein that's associated with those receptors. So the receptor itself is this 7-pass transmembrane protein that we talked about before. You've seen a hydropathy plot of a 7-pass transmembrane protein, and the idea here is that it goes through the membrane 7 times. It has an extracellular region that binds to ligand. It has an intracellular region that causes the recruitment of the G protein linked receptor molecule. Okay, sometimes we call it a serpentine, right? A serpentine means like an ass or a serpent, a serpentine receptor. And the G protein, which is shown in panel A to the right, is actually made up of 3 different proteins, an alpha, beta, and gamma subunit. Notice that the alpha subunit in this case is linked to the membrane. It has a lipid modification to it that allows it to stay linked to the membrane, as well as the gamma subunit also has a link to the membrane. So those molecules, they're going to be recruited to the changing conformation of the cytosolic portion of the trimeric 7-pass transmembrane receptor, and they're going to, even though they're going to dissociate from one another, they're still going to stay bound to the membrane. They can't diffuse freely throughout the cell. So ligand binding changes the conformation, and that's shown by the little red line saying, hey, I've changed my shape and now I'm active. The binding of the G protein, okay, so the whole alpha, beta, gamma subunit, the inactive G protein, is recruited to the receptor. When it binds to the receptor, it causes a change in conformation that allows for the release of GDP from the alpha subunit. That allows GTP, which is way more prevalent in the cytosol than GDP, to bind. That GTP binding to the alpha subunit causes a change in conformation to the alpha subunit, causing the release of the beta gamma. And notice they're released from one another, but they're both still bound to the membrane, and they signal. And those signals, right, remember the beta gamma complex signals, even though we don't know what that signal is. I know that it now does something. We used to think it was worthless, but the regulation of this signal, right, is all going to be dependent on the hydrolysis of GTP. How quickly does GTP get hydrolyzed? Once GTP gets hydrolyzed and GDP is around, the beta gamma subunit is going to come back and bind to it. And now we're going to turn off that signal. So this is really the same exact thing that we talked about before with all of the other GTPases. We talked about RAB, we talked about dynamin, we talked a little bit about SAR and ARF. Okay, all of these molecules, so RAB, RAC, RAS, RAN, RHO, these are GTPases that bind GTP, changes their conformation, activates them, causes the recruitment of what we call effector molecules to the activated RAB. The hydrolysis of GTP, once again, changes the conformation, thereby allowing for the dissociation of effector molecules. And remember, that's all regulated by GAPs and GEFs. GAPs accelerate the hydrolysis of GTP, whereas GEFs allow for the removal of the GDP to allow GTP to bind. There is a specific gap for G proteins. I don't care if you know this, okay, but it's called the regulator G protein signaling, RGP. All right, the beta gamma subunit, right, is waiting in the wings, doing whatever it is, but we do know that when it binds back to the GDP-bound alpha subunit, the complex has basically been recycled back to its original position, waiting to be activated again by binding of ligand to the receptor, change of the conformation of the cytosolic domain, and recruitment of the inactive G protein. Remember, there's 3 different classes of G proteins. There's GS, which stimulates the production of cyclic AMP. There's GI, which inhibits or decreases the production of cyclic AMP. And GQ is the weirdo. GQ actually activates phospholipase C, which leads to the production of diacylglycerol and IP3. Cyclic AMP is really no different than the formation of cyclic GMP. In this case, you have an adenylyl cyclase that's activated. So when we're talking about the alpha subunit of a GS protein, the alpha subunit is going to somehow activate the adenylyl cyclase. That could be by direct binding. I don't really know, to be honest with you. But the activation of the adenylyl cyclase causes the formation of cyclic AMP from ATP. The energy from the 2 phosphate bonds at the end of ATP are used to generate the cyclic molecule, and cyclic AMP has the activity downstream. So cyclic AMP is the second messenger in this case. The removal of cyclic AMP, similar to the removal of cyclic GMP, is regulated by a phosphodiesterase. Phosphodiesterase comes along, cleaves that bond between the 3' hydroxyl, right, and the 5' phosphate group, resulting in the formation of 5' AMP. And what that really is we've just reset the system back to nothing. The proteins that I talked to you about as far as GS subunits and GI subunits, the one that I always find interesting is cholera. So cholera causes severe diarrhea, and severe diarrhea is through the activation of GS subunits in your epithelial cells of your gut. So in your intestines, if you have this bacteria, the bacteria actually secretes a toxin that activates the GS subunit on your epithelial cells. The increase in cyclic AMP in your epithelial cells causes chloride influx and water influx into the lumen of your intestines. That's bad because, right, water in your intestines is just going to give you runny diarrhea. And unfortunately, it's really difficult to inhibit that toxin. The best way to get rid of this is to flush it out. So the way people, right, if you get diarrhea, the best thing you can do is keep well hydrated, drink lots of fluids, just keep the fluids coming, because you're basically going to wash out the toxin and hopefully clear the bacteria. You need to wait for your system to clear the bacteria, or you need to take an antibiotic to get rid of whatever that bacteria is. Pertussis toxin, on the other hand, whooping cough, right, that's caused by an inhibition of the GI subunit. okay, in the lungs. So a GI is supposed to inhibit the formation of cyclic AMP. The inhibition of a GI subunit causes an increase in production of cyclic AMP. In this case, the production of cyclic AMP leads to this coughing due to, I'm not exactly sure what it is, whether it's an increase in mucus production or whether it's just an increase in some sort of irritant on the internal linings of the lung itself. This is what whooping cough is. This is also what kennel cough is caused by another form of Bordetella caninensis or something like that. So G proteins really do make a huge difference as far as, what we end up with nowadays. Pertussis toxin, Bordetella pertussis, huge thing, right? We now need to get revaccinated for whooping cough because it's becoming a severe problem, especially in, I know that in Seattle there's a huge outbreak right now. So, and it can be deadly to small infants. Okay. Well, where do we go from there? What the heck does cyclic AMP do? Cyclic AMP does a whole bunch of things. It's a second messenger. It's going to have multiple functions. The multiple functions can cause any number of downstream events. One that we talked about was cyclic AMP dependent protein kinase A. So cyclic AMP can bind to the regulatory subunits that are bound to protein kinase A in the cytosol, right? And when that happens, it causes the release of protein kinase A, and protein kinase A is a serine threonine kinase, and it's going to go phosphorylate other proteins. When it phosphorylates other proteins, right, it can activate them. So in this case, we're talking about a phosphorylation event that can activate or inhibit other molecules. Some of those responses to protein kinase A, right, have to be really, really fast, right, for things like glycogen metabolism. During skeletal muscle contractions, okay, you can actually activate the production of cyclic AMP to then help you release glycogen. So glycogen is nothing more than a alpha-1,4 linkage between a bunch of glucose molecules. And glycogen can be quickly broken down when protein kinase A gets activated. That has to happen really quickly for you to be able to use the glucose molecules immediately to generate energy for skeletal muscle contractions. Protein kinase A can do all sorts of really slow-acting activities too, which we talked about. Cyclic AMP activating protein kinase A, which leads to the phosphorylation of this cyclic AMP response element, and CREB is the cyclic AMP response element binding protein. If you look at that, okay, cyclic AMP binds to the regulatory subunits, releasing pKa. pKa goes to the nucleus, binds and activates CREB, and CREB then binds to the promoter sequence, leading to gene transcription. Anything with gene transcription, slow, okay? So depending on what is going on, this cyclic AMP can lead to very quick reactions or it can lead to very slow reactions. Okay, that leads us to the GQ protein. GQ protein gets activated. Okay, this is just a different ligand binding to a different G protein that's going to recruit a different alpha subunit. This alpha subunit is now, when it gets activated by binding to the 7-pass transmembrane receptor, is going to then cause the activation of phospholipase C. Phospholipase C cleaves PIP2. PIP2 is a phosphoinositide that's found on the inner membrane of the plasma membrane. When it gets cleaved, it produces a soluble product, which is called IP3, or inositol triphosphate, and that can freely diffuse through the cell, and you're going to see that's going to activate the release of calcium and diacylglycerol. Diacylglycerol still has the fatty acid chains that linked the phosphoinositide to the plasma membrane, and it has a diacyl, right, glycerol molecule still attached to it. So this is a molecule that's going to remain bound to the membrane, but it also causes downstream signaling. So IP3 is really important. We know a lot about IP3, and we can mimic the activity of IP3. And this actually binds to a calcium channel on the ER, and it causes the release of calcium from that reticulum. The reticulum can either be the smooth ER or the rough ER. Sometimes it's called the sarcoplasmic reticulum, depending on the cells. This is not the same thing as the activation of the sarcoplasmic reticulum in muscle cells. This is a different activation mechanism, completely separate, nothing to do with muscle contraction. Okay, so that causes the release of calcium. Calcium is then going to go bind to things, right? There's lots of molecules that bind calcium, causing a change in conformation, changing their function of whatever those proteins are. Diacylglycerol, on the other hand, is going to then activate protein kinase C. Okay, this is just another protein kinase, right? We have protein kinase A, we have protein kinase B, we have protein kinase C, lots of different protein kinase, serine threonine kinase, causing some sort of downstream signaling. So if you look on this, right, this chart, okay, if you know what G proteins, so the vast majority of G proteins, GS and GI, are all about cyclic AMP, right? So you activate an adenylyl cyclase, you make cyclic AMP. Cyclic AMP activates protein kinase A. Okay, so that's the first thing. That causes all sorts of different target proteins to be activated. GQ activates phospholipase C, causes diacylglycerol and IP3.